Hello, students. Today we're going to talk about citric acid cycle, which is also abbreviated as TCA, and then it's also known as Krebs cycle. And we looked last time at glycolysis and gluconeogenesis, and so we're going to review a little bit on especially on glycolysis today, talk about the fates of pyruvate, and then talk about citric acid cycle and its importance. So glycolysis as a review, remember it's carried out in all cells in the cytosol. It has 10 reactions, and it has two phases. One is the preparatory phase that requires ATP, and then you have the second phase, which produces two pyruvates and 4 ATP, and for a net gain of 2 ATP. And the main products are pyruvate, ATP, and then NADH. Let me show you some structures for NADH. NADH is what's called an electron carrier, and it was formed in glycolysis by gaining 2 electrons and 2 H plus to form NADH. In citric acid cycle, we're going to see another species, another electron carrier called FADH2. So I just wanted to show you their pictures because that wasn't in the last PowerPoint. Here's NAD, and it has a fancy name, nicotinamide adenine dinucleotide. One other thing I want to start bringing up is the importance of vitamins. So niacin, which you might have heard of as a vitamin, it is a precursor to this. And so vitamins play such a critical role in metabolism, but they're support players. They tend to be building blocks for some very important key molecules. And so I think as nutritionists and dietitians that the importance of diet goes beyond just how people look and are they getting what they need to be healthy. And so vitamins for sure are important in metabolism because they're the building blocks of some very important molecules. So niacin is the building block of NAD. Now it's a very complex structure. I think you can appreciate, very interesting. And so on the next slide, we're going to condense it a little bit so that it's not so intimidating. And so you notice this hydrogen here with the double bond. Okay, it is going to gain H plus and 2 electrons. So that is a reduction. Now, where is it getting this from is that it'll be in conjunction. If this is getting reduced, something else in the process is getting oxidized. So reduction, recall, is the gain of electrons. Here's FAD, not seen in glycolysis, but that we will see in citric acid cycle. Wow, what a weird looking molecule. Its vitamin precursor is riboflavin, which is vitamin B2. And so again, the importance of vitamins. And so let's take a look at the structure in an abbreviated form. The R group is the rest of all that stuff. And then if you notice this nitrogen here is going to gain H plus and 2 electrons within the structure. There's some rearranging, right, of electrons within the rings and forms FADH2. So again, the gaining of electrons is reduction, and this would be in a process where something else is being oxidized. Okay, so here's our glucose going to pyruvate. So just a review of glycolysis. So in phase 1, you have ATPs required, so you need energy. Remember, it's a 10-step process. So step 1, 2, 3, and then all the way, I won't recount them, but here's step 10. And steps 1, 3, and 10, if you recall, are highly exergonic. And step 10 in particular is so interesting because it's going to pull through the reactions in between here that are in an equilibrium state and kind of are reversible. So it's going to pull them forward. When would this happen is when there's a demand for ATP. So remember, some of the enzymes are regulated where if you have high amounts of ADP, which is meaning you need ATP, then glycolysis is activated, whereas high amounts of ATP inhibit. The other thing just to highlight here, here's our ATP, ATP, okay, NADH. Now, why is this showing in double, right? Why is it on both sides? Is if you recall that the one step produces from fructose 1,6-bisphosphate produces dihydroxyacetone phosphate and glyceraldehyde 3, and then dihydroxyacetone phosphate is converted to a second glyceraldehyde 3. So it's going through twice. So this is why you end up with 4 ATP. You need 2. So our net amount is 2 ATP. And then here you have 2 NADHs. So at the end of the day, in glycolysis, you have 2 pyruvate, 2 NADH, and 2 ATP. And now the question is, what happens to the pyruvate? At the bottom of this slide, it gives us 3 possible options, and then I'm going to go through them also on the next slide. One is for us kind of the least important, which is fermentation in yeast, because we're not yeast. And then there's the one that is for energy pathway, which means it's going to enter into citric acid cycle. And then, or under anaerobic conditions, it can go and make lactate. So let's talk about that on the next slide. It's just going to be easier for me to write. So here's pyruvate. So let's just get it over with and talk about the yeast. So this is fermentation. If you've ever made like beer, sometimes students like to make beer, you will make, you'll convert sugars, glucose sugars, into pyruvate, and then they form ethanol in the presence of yeast, and then also form CO2, the carbonation. Okay. Now, in animal cells, there's 2 possibilities. One is in muscles, where you have low or no levels of O2, so in strenuous activity where that's being depleted. What will happen is pyruvate's converted to lactate through lactate dehydrogenase, the enzyme. And lactate then creates that burning sensation, right? The burn that you feel from strenuous muscle activity. Now, there's some other side notes here that are super important. One is that, see how it reforms NAD+? That's going to be really important because you need to regenerate NAD+ for glycolysis, right? We just saw that we make NADH. So that's a way, this process is a way of regenerating NAD+ to keep glycolysis going. And then another thing that happens here, not shown, is that lactate ultimately can go to the liver. And in the liver, this can be a substrate for gluconeogenesis. So we'll talk more about pulling all the pathways together towards in a couple weeks, but keep that in mind that, and I'm going to bring up gluconeogenesis again because the pathways are all interlinked. And so one way that we can make glucose is from lactate. Okay, now we have for today's discussion, what will happen. Let me come up here one thing before I finish. Strenuous muscle activity, O2 depleted. Sorry, I should add that in. That looks funny like that. So that's why this is considered to be anaerobic without oxygen. In aerobic conditions, you're going to see that pyruvate is converted to acetyl-CoA, and this is when there's a demand for energy. And so we'll come back to the nitty-gritty of this step. This is a pretty famous enzyme called pyruvate dehydrogenase, and it has a lot going on. And we'll see some steps a little bit later as well, but I could just introduce it right now. You have a very famous molecule called coenzyme A. Oh boy, that was not good. Did you do that? I think I have highlighter on there. Let me try to erase that guy. So this is coenzyme A. We'll see a pic here in a few minutes. NAD plus is required to make NADH. And then notice there's CO2. Now this is our friend CO2. We're going to start seeing more of it. This is what we exhale, right? The carbon dioxide we exhale. We talked about how it changes the pH of the blood. It binds to hemoglobin. So this is where it's going to come in, or this is where it's formed, one of the spots. Okay, so here is the idea of the conversion of pyruvate to acetyl-CoA. And one of the interesting things is that acetyl-CoA, as we keep moving forward, is what I consider to be the hub of metabolism. And so it could go into energy, which is what we're focusing on today, or it could also be stored as fat. All right. Okay, so let me skip this one, just to pick another picture. Okay, so glycolysis is in the cytosol. Krebs, or citric acid cycle, its abbreviation TCA, is in the mitochondrial matrix. And so the mitochondria are a specific organelle within the cell, and it has an outer membrane that's very permeable, but then it has an inner membrane that is more selective. So it is permeable to pyruvate, but impermeable to fatty acids and NAD. And the matrix is inside of the inner membrane. So I think you need to pick, right? So here's a mitochondria, and it has the outer membrane. It has an inner membrane. And then, okay, so the blue is the matrix, the middle part. And then in between the outer membrane and inner membrane is what's called the intermembrane space, which is highlighted here. Okay, these folds, how it kind of folds, are called the cristae. And this is a picture of it from a very fancy scanning electron micrograph or microscope. Okay, so as we think about this, then there's going to be, kind of in a more simplistic view, here's the outer membrane. Here's the inner membrane. Here's the matrix. And then here's the intermembrane space. And try to keep that straight, because it's going to be a discussion for us to kind of know where things are happening. Okay, so going back, let's just go back to the previous slide. citric acid cycle will happen in the mitochondrial matrix, whereas glycolysis is not even in the mitochondria, but it's in the cytosol. So picture that this is part of a bigger cell, right? Glycolysis is out in here. And then in the matrix, the blue part is where citric acid cycle happens. So the pyruvate has to enter in, as does the NAD. Okay. Okay, so this is a prettier picture of what I just tried to draw. So here's the cytosol of the cell. Here's glycolysis, glucose to pyruvate. Pyruvate has to enter and go through both of the membranes to get into the matrix, and the electrons get carried via NADH in there as well. And ultimately, we'll keep going and talk about electron transport chain and how that's going to be a major way of making ATP, but we're getting ahead of ourselves. So what I just want you to realize is the location that these things are happening at. Okay? All right. So the overall goal of citric acid cycle is to make ATP, make NADH, and make FADH2. Now, I would also tell you at the end of this discussion, we're going to see how important citric acid cycle is to other pathways. So it goes just beyond this idea on this slide. So we'll pull it together at the end. Okay, so remember when I told you that the enzyme is super famous? So our pyruvate dehydrogenase, that wasn't a good color. So pyruvate dehydrogenase, this is a really important enzyme, and it's also an incredible enzyme. Super complicated. I know I'm going to show you some slides on how complicated it is, just so that you can marvel at how amazing it is. But you don't need to know the details. I just want you to see what an incredible enzyme it is. It has it's called a complex. It has 3 different enzymes: pyruvate dehydrogenase, dihydrolipamide transacetylase E2, and then dihydrolipamide dehydrogenase E3. And so those are 3 different enzymes. So it's called a complex. The other thing that I want to tell you is that within those enzymes, there's a bunch of cofactors and coenzymes. And I think it's important to see how important vitamins are. So we're going back to the vitamin discussion. So one of the cofactors in this big monster enzyme complex is thymine pyrophosphate, TPP. And thymine, a precursor to thymine, is vitamin B1. So vitamin B1 is thymine, and then thymine is within that cofactor. Okay. See here, FAD, we already talked about how riboflavin, vitamin B2, and niacin for NAD. So I'll just keep pointing out the vitamins, how they play really important roles in the enzymes, even though they're not the substrates themselves. Okay. So pyruvate to go to acetyl-CoA is going to need NAD+, and this is just coenzyme A. And then it goes to acetyl-CoA, NADH, and CO2. And then this is the CO2 that we exhale. And this is a very committed step, meaning that it releases a lot of free energy. So it's a one-way deal here. This picture is just for you to be amazed as to all of the different complexes that make up this monster enzyme structure. And so it's huge. And so anyways, just hopefully you can see that it's pretty amazing. And then you don't need to know. So you don't need to know the specifics. So I'll just put a little X there. But this is just for you guys to see. You know, we spent a lot of time on serine mechanism, and I think you did really great on that. This is just another mechanism. And I won't go through everything, and I'm just going to show it to you. But it just goes to show you how each enzyme will have a different mechanism and different cofactors, different electron transfer reactions, different types of processes. So serine protease was one type. This one's very different. You can see it looks different. Anyways, it's just to show you that it's a very complicated step within metabolism. Oh, the other thing too is, okay. No, I was going to say something else, but it's okay. So coenzyme A, here's coenzyme A, another kind of interesting complex molecule. What I want to show you on this is that coenzyme A also has a vitamin precursor. And this one is pantothenic acid or vitamin B5. And coenzyme A is so important in our metabolism. It's going to get abbreviated a lot of times like this, just CoA, or it's going to focus on this part, the sulfur part. You see that? So be aware that when it's abbreviated like that, it's just trying to abbreviate this last part, which is the sulfur part, which is the reactive part of this. Okay, so I love this slide, super important. And basically, it goes to show you how important acetyl-CoA is. And I probably should have on the previous one, coenzyme A, remember, was like this. I just showed it to you as a abbreviation. Whoops. Let me go back on that because we just had it up there. So this was CoA. And then let's go back even a little bit more since I can easily. Okay, let's go back here. Here's CoA. In the process of reacting with pyruvate, okay, and NAD, the CH3 group is transferred from pyruvate onto CoA to make acetyl-CoA. Where's the acetyl part? From your OChem, it's the CH3 part. Okay, so hopefully that's clear. And now let me click now on the next slide. Oops. Okay, so you can see that the transfer, and I didn't highlight it correctly on that previous slide. Let me go back. It might have not been showing, or it's just my oversight. Hold up. Here, I needed to extend that to not just the methyl group, the acetyl groups here. Sorry, sometimes talking, highlighting, clicking. Okay, so that's the acetyl group that's been transferred. Okay, so there's the acetyl group of pyruvate, and then you see how it ends up on the CoA here. So this group here is the reactive one. And then you see how there's that sulfur, and now it has the acetyl group on there. So when you are looking at these, if you just want regular CoA, it's written like this. And acetyl-CoA is written like that. So this is just CoA, and this is acetyl-CoA. Now, I love this slide because it just shows us how important acetyl-CoA is to metabolism. It can be stored as fats, so in excess amounts. Can be stored as fat. So TAGs, triacylglycerols. If we need energy, acetyl-CoA will go into Krebs or citric acid cycle to form ATP, NADH, and CO2. And then if we don't have carbohydrates present, it'll form ketone bodies. And we're going to talk more about that a little bit later. Okay, that's when you might have heard of the term ketosis, and it's really popular. It's been popular for many years now, but we're going to talk about pros and cons, but not at this point. Okay. All right. So this slide, it's simple, but I think it shows us all the possible things that could happen with acetyl-CoA. And it's really the hub of metabolic processes. Which way this goes will depend on hormonal signals. It'll depend on what's going on with carbohydrate levels in the body. And so it's pretty complicated which route it picks. And when we pull this all together, that's what I call the integration of metabolism. Okay, and we'll get to that. Okay, don't let this scare you. What have we done here, and what is this about? What I want you to think about is that we just talked about pyruvate going to acetyl-CoA, and that acetyl-CoA is the hub of metabolism. Later, when we talk about fatty acid metabolism, the breakdown of fats for energy, this is where fats will be broken down for acetyl-CoA. Now, acetyl-CoA had those fates we just looked at. This one is just focusing on when we have energy demand. So when there's an energy demand, it's going to go this way. It's not concerned with those other demands, other possibilities right now. Interesting side note, and I'm going to bring this up right now, is that amino acids can form and come into play to be broken down for energy, but this is not a desirable thing. And so what will happen is there is an arrow there, but we're going to look at the metabolism of amino acids and under what situations will it get pulled into these pathways. Okay, so we'll come back to the amino acid discussion. I know amino acids and proteins are very popular in your generation, but they're not necessarily designed to be key energy molecules. Okay, now citric acid cycle is a cycle, so it goes all the way around. So the other thing here is that, remember, you're going to have two pyruvates, right? So you're going to have, for every glucose, you can have two pyruvates and you have two acetyl-CoAs. So citric acid cycle will churn twice for every glucose. The other key highlights, if I was to say what are the key highlights are, is that you'll see there's CO2 comes off. So a lot of the CO2 we breathe out comes into play as a waste product from citric acid cycle. Another thing is that you do have a spot where you form ATP, and for some reason, not shown, well, I guess it's indirectly shown, you actually will make 3 NADHs for every time you churn around. So 3 times 2 for the 2 acetyl-CoAs going around. So the NADH is formed and also will form 1 FADH2 times 2 because it's churning around twice. And here's the big picture. is our next pathway is called electron transport chain. Those electron carriers are going to enter into that pathway, and that's where we're really going to see a lot of ATP formed. Okay. So I kind of like this picture a little bit better. So here's our pyruvate dehydrogenase, formation of acetyl-CoA. See where it says beta oxidation from fatty acids? We'll do that soon. That's going to be pulling fats for acetyl-CoA. So if we need more energy from fats, then those are going to come into play here, and then this goes around. Okay, notice some important steps. You have NADH is formed in 3 steps. So that's why I mentioned to you that you have 3 NADH formed for each pyruvate. So for 1 glucose, you make 2 pyruvate. 2 pyruvate make 2 NADH. From pyruvate dehydrogenase to make 2 acetyl-CoAs. And as this churns around, you make 2 times 3 NADH. You make 2 times 1 FADH2, which is here in that step. And then you also will make GTP, which we talked about with the G protein cascade. It's that same guanine triphosphate. And then that gets converted to ATP. So you make 2 times 1 ATP. So this is what I'm going to call our bookkeeping. So if you could write that down somewhere, that's going to be helpful a little bit later when we come back and start talking about the final energy that we can get from 1 glucose. Okay, a couple other things is that CO2 that we breathe out is given off by citric acid cycle. Okay, and if you're worried that you'll have to write these steps, you don't. You know, it's just understanding what are the key features of the pathway. Okay, here's another picture. Okay, I won't rewrite it, but if you like this graphic better, that's fine. Okay, so quickly, you're not responsible for the formulas. It's just more understanding what's happening. So acetyl-CoA will combine with oxaloacetate. Oxaloacetate is very, very important because it is going to be found in gluconeogenesis.
You want the tortellini or the Parmesan?
And so it can be diverted to make glucose if we need it. And if it's being diverted from citric acid cycle, what that means is you're going to slow down or shut off Krebs. Okay. All right, but in this case, we're assuming that that's not the case. So I'll say can be diverted to make glucose if needed. Now we're assuming that that's not the case, that we're going to move forward for energy. So acetyl-CoA will react with oxaloacetate to form citrate. Very, very exergonic, so very committed at this point to moving forward. Citrate will go to isocitrate, okay, and this goes through an isomerization and addition of water, release of water. So here, it's essentially reversible. If you notice the delta G's around 0, and so it just means it's a reversible dehydration, and addition of water. This step here, isocitrate dehydrogenase reaction 3, is where we get the formation of NADH. So the NADH is reduced. And remember, if something's reduced, something else has to be oxidized. Okay, so in this case, it's going to form alpha ketoglutarate through this intermediate of oxalosuccinate. So this is the intermediate thing there. When you form and release CO2, that is called oxidative decarboxylation. So that means that CO2 is given off in that step. And then that's the one that we breathe out and goes through our bloodstream, etc. Reaction 4, alpha ketoglutarate to succinyl-CoA. Again, it's driven by the loss of CO2. So you have an entropy discussion here. What's going to propel this forward? Because this would be a very difficult reaction to do. But the loss of the gas, remember we said from our previous discussions, in biochemistry, this term of enthalpy is not as important, that it's processes that increase the entropy that tend to have a negative delta G. And so this is a great example. Now I can give you a more concrete example that the formation of CO2 gas is an increase in entropy. And it's driving this to be exergonic. Succinyl-CoA to succinate is a exergonic reaction that is coupled to the formation of GTP. So then GTP will interconvert to ATP in a side reaction. So think about this, that the formation of GDP, this requires energy. And that energy comes from the hydrolysis of the thioester. So here, this break in that bond to form the to reform coenzyme A, that releases energy. And I love this because this is an example of driving an endergonic reaction with an exergonic one. So for sure, this is an example of reaction coupling. Succinate dehydrogenase. This is one I'm going to bring up to you a little bit later in our next video. It is actually part of the inner mitochondrial membrane. So it's an integral protein. It requires it requires and uses FAD, and that's actually bound to that enzyme. And so this is a step that will form FADH2. Okay, but we're going to see this one again in electron transport chain. So I'll make a little note of that here. It's going to come back up there. The addition of water here to this double bond takes fumarate to malate. And then malate reforms oxaloacetate, takes a secondary alcohol to a ketone. It regenerates oxaloacetate for another round and makes NADH. Okay, but I do want you guys to make a note of our friend oxaloacetate because it's going to play a role when we start integrating these pathways together. Okay, so here were our numbers that it makes 3 NADH, 1 FADH2, and 1 GTP per acetyl-CoA. So it's churning around twice though. Okay. And later, the electron carriers of NADH and FADH2, these guys are going to be where we generate a lot of ATP. So that's in the next section. Overall, Krebs cycle releases a lot of free energy. Okay, so it wouldn't make sense that you need energy to ultimately make. So it has that driving force is once it's committed into this pathway, it's going to go make these electron carriers to help make more ATP later. Okay, so take a minute to do some bookkeeping now and put this somewhere in your notes that where we at in the cytosol, we have glycolysis. These are the numbers. Okay, the mitochondria, pyruvate dehydrogenase, that was the reaction of pyruvate to acetyl-CoA gives 2-NADH. Up here, this was glucose to pyruvate. And then Krebs churned around twice to make these. So this is the ultimate goal of Krebs cycle, and it's not showing the CO2, that's a waste product. Okay, so keep track of this because then we're going to talk about this in terms of our next pathway, which is how do we convert NADH and FADH2, those electron carriers, how do they play a role in making more ATP? Okay, so one of the big themes is how do we control this pathway? This pathway would be controlled in those key steps. And if you look closely at the diagram, really the biggest step is the conversion of pyruvate to acetyl-CoA. That if we have high amounts of pyruvate, that's going to push it forward. If we have high amounts of ADP, meaning we don't have enough ATP, it's going to push it forward. Insulin will play a role. We'll still talk about that piece later. So this was a super exergonic step. And so the regulation point then is there, which makes sense. And then there's some other regulatory points, but this is our main one, is controlling that particular point, pyruvate dehydrogenase. Okay. Oh, another thing too is what also can occur is that your ratio of NAD plus to NADH also controls this. So if you have too much NADH around, right, too much, then that's going to shut this down because you don't want to be wasteful, right? So if you have high amounts of NADH and it's not needed to convert to ATP, then it's going to regulate this negatively. So this is positive regulation on this side, pushing it forward, and this is essentially inhibition. So I think it makes sense that high amounts of acetyl-CoA going to shut this down, high amounts of NADH, and high amounts of ATP. So if you have those energy molecules, high amounts of them, you're not going to want to make more of them. Okay, I'm going to put a little heart next to this one as well, because I want to start to get you to think about how these pathways, especially Krebs, is linked to other pathways. So here's our Krebs cycle. Here's our important oxaloacetate that I really like discussing, is that, let me show you, there's this arrow, oxaloacetate to phosphoenol pyruvate. See that arrow here? Oh, I don't know if I can draw really well. I have to really focus. Ooh, wobbly. Okay. That is gluconeogenesis. go back to that video and look at that part. Oxaloacetate to phosphoenolpyruvate is a step of gluconeogenesis. It was that one step that had to change because, remember, here, let me look at it this way. Phosphoenolpyruvate to pyruvate, that's step 10 of glycolysis. But it is super exergonic. So to go backwards in gluconeogenesis, we can't, because this delta G was so negative, it's a forward only. So what there was that was one of the bypassed steps. Pyruvate to oxaloacetate to phosphoenolpyruvate. That's gluconeogenesis. Now, why is this so important? Oh, I spelled that a little wrong, sorry, but okay. This is important because later when we talk about situations where you haven't eaten for a long time, your body's not going to care about getting energy for your muscles. It's going to want to preserve your blood sugar levels and your brain. And so what will happen is oxaloacetate will get diverted out of Krebs and get pushed into gluconeogenesis. You see how this is gluconeogenesis to making glucose. This will go into the bloodstream and go to the brain. And I don't know if you remember, gluconeogenesis predominates in the liver. Okay. All right. Oxaloacetate is also formed from some amino acids like aspartate. So again, if you haven't eaten, if you're in a very extreme in starvation, that's where you're going to start pulling from proteins to make glucose for the brain. So aspartate can be converted to oxaloacetate, which can then go into gluconeogenesis. Okay. Other things shown here, which I won't stress too much on, are citrate can go to make fatty acids and sterols. Succinyl-CoA can form heme and chlorophyll. The one, though, that I really like is alpha-ketoglutarate to glutamate and form other amino acids. This is going to come up for us later. Transamination is an important process in the body because as we process amino acids, when amino acids are processed, they give off ammonia, and that's toxic. So there has to be a way of transferring that ammonia onto other molecules so that ammonia doesn't free float in the blood and kill us. And that's transamination. It's the transfer of ammonia groups. Okay, so I know there's a lot going on here, and we will come back to this, but I just want you to understand that pathways are all linked. So up here, this was glycolysis, right? Which I know you've learned in other classes, but I'm guessing, you can tell me in the Zoom, that sometimes they don't necessarily mention gluconeogenesis and the role of Krebs cycle in that. But that's a key regulatory feature in our bodies, especially when we're low in blood glucose, like if we haven't eaten in 4 to 5 hours, and becomes enormously critical in prolonged fasting or starvation.